Now let's look at how helper T cells aid cell-mediated immunity and humoral immunity. Imagine that bacteria have infected the body. Antigen molecules on their surfaces identify them as foreign. A macrophage engulfs an invader and breaks it into pieces. It displays the foreign antigen on its surface, along with a self-protein that acts as a label for the body's own cells. The macrophage is now an antigen-presenting cell. Helper T cells are key components in the immune response. There are many kinds of helper T cells, each with a unique kind of T cell receptor. Each receptor binds and responds to a particular self-non-self combination of antigen and self-protein, making the immune response highly specific. Binding of the T cell receptor to a particular antigen and self-protein activates the T cell. A chemical signal, interleukin-1, enhances this activation. The antigen-presenting cell secretes interleukin-1. Both interleukin-1 and contact with the antigen-presenting cells activates specific helper T cells to divide and grow into many cells with the same receptors. The helper T cells next activate other cells in the immune system by secreting another stimulatory protein, interleukin-2. Interleukin-2 stimulates helper T cells, cytotoxic T cells, and B cells. The helper T cells divide more rapidly and increase their stimulation of other cells. Cytotoxic T cells are stimulated to become active killer cells. B cells are stimulated to develop into plasma cells that secrete antibodies against the specific antigen.